Welcome to The Explainer. Today, we're taking a fascinating, literal zoom-in journey through one of the most complex structures in the known universe, the human nervous system. To put this into perspective, you have enough microscopic wiring inside your body right now to wrap around the Earth multiple times. No, seriously. We're going to start wide, looking at the macro-level hardware of the body, and then systematically zoom all the way down to the micro-level software, the actual electrical impulses and chemical signals that make you, you. It's this incredible system where biological tissues seamlessly generate and conduct electricity to coordinate every single thing you do. So, let's dive right in.
Here's our roadmap for today. We'll be mapping the nervous system, checking out neuron anatomy, meeting the support staff, the neuroglia, and then diving into the resting potential, the action potential, and finally the synapse connection.
All right, part 1, mapping the nervous system. We're starting at the absolute macro level to map out the broad anatomical divisions of our biological hardware.
The big thing to grasp here is the main divide. On one side, you've got the central nervous system, or the CNS. This is your brain and your spinal cord. Think of this as your ultimate processing center. It contains the neural tissue, connective tissues, and blood vessels responsible for processing sensory data, sending out motor commands, and handling all those higher functions like memory, intelligence, and emotion. On the flip side, we have the peripheral nervous system, or the PNS. This includes all the neural tissue outside of the CNS. The PNS is essentially the vast network of cables, your nerves, delivering sensory information to the CNS and carrying motor commands back out to your peripheral tissues.
So, let's break that vast peripheral network down a little further. The PNS splits into these dedicated functional delivery routes. First, you have the afferent division, which brings sensory information from outside receptors into the CNS. Then, the efferent division carries motor commands from the CNS back out to your muscles and glands. Now, this efferent side splits again into the somatic nervous system, which handles your voluntary skeletal muscle movements, and the autonomic nervous system, which governs all the involuntary subconscious stuff like smooth muscle contractions and glandular secretions. And as a final layer, that involuntary autonomic system constantly balances between the sympathetic division, which hypes you up and expends energy, and the parasympathetic division, which relaxes your body and conserves energy.
Moving right along to Section 2, the neuron anatomy. Now that we have our macro map, let's zoom in closer and look under the microscope at the individual wires of this network. Form absolutely follows function here. The neuron is the basic functional unit of the nervous system. It's responsible for all communication. Signals come in through these highly branched dendrites, which, by the way, make up something like 80 to 90% of the neuron's surface area. They act like massive antennas receiving information. That signal then passes through the main cell body, the perikaryon, which houses the nucleus and these dense areas of cellular factories called rough endoplasmic reticulum. Those factories actually make the tissue appear gray, which is where we get the term gray matter. From there, the electrical signal shoots down a single long structural cable called the axon to reach its ultimate target.
But you know, not all neurons rock that classic shape. They come in different physical forms to tackle very specific jobs, falling into 4 main structural categories. For instance, you have multipolar neurons, which are super common in the CNS and include all the motor neurons controlling your skeletal muscles. But compare that to a unipolar neuron, where the cell body just sits off to the side. Those are typically found in the sensory neurons of your peripheral nervous system. Then you've got bipolar neurons, found in special sensory organs like sight and smell, and anaxonic neurons found in the brain. Their structural diversity is literally what allows them to handle their unique processing roles.
All right, Section 3, neuroglia. Neurons get all the glory, for sure, but we really need to shift our focus to the support staff that actually makes their work possible.
Neuroglia, or glial cells, are basically the janitors, the bodyguards, and the insulation experts of the neural world. While neurons are doing all the flashy firing, they rely entirely on neuroglia to preserve the physical and biochemical structure of the tissue. And get this, these unsung heroes actually make up half the total volume of your nervous system. Without them, the entire communication network would just totally collapse.
It's wild to see how complex this support network is inside the central nervous system. You have all these different types of cells clustered around the neurons. There are astrocytes, these big cells with many branches that are absolutely critical for maintaining the blood-brain barrier, basically isolating the CNS and controlling its internal environment. Then you've got ependymal cells lining the central canal, using tiny cilia to secrete and circulate cerebrospinal fluid. And let's not forget the microglia, which migrate through the tissue, acting as a cleanup crew for cellular debris and pathogens.
It's this highly coordinated ecosystem dedicated entirely to keeping the neurons functioning perfectly.
And over in the peripheral nervous system, it gets even cooler with a specific type of glial cell called a Schwann cell. These cells literally wrap their entire body around the axon of the neuron to form a myelin sheath. This myelin acts as vital electrical insulation. By insulating the axon, leaving just these tiny gaps in between called nodes of Ranvier, myelination massively increases the speed at which electrical action potentials travel. We're talking up to a staggering 120 meters per second. It also gives these specific nerves a distinct white appearance, which is exactly what we mean when we talk about white matter.
Section 4. The resting potential. So we understand the biological hardware and its insulation. Now let's turn on the software and talk about electrical charge.
I want you to focus on this exact number, -70 millivolts. All cell membranes produce electrical signals by moving ions around, maintaining a charge difference across the membrane. This specific number, -70 millivolts, is the resting transmembrane potential of a neuron. I love this concept. It is the literal threshold of human consciousness, just a biological battery fully charged, waiting in the dark, primed to fire at a moment's notice.
But how on earth does a living cell hold that exact negative charge? Well, it's a constant tug-of-war between passive and active forces. Ions, like sodium and potassium, want to passively leak across the cell membrane. To counteract this, the cell uses a brilliant active mechanism called the sodium-potassium exchange pump. Powered by ATP, which is the cell's basic energy currency, this pump is constantly working, shoving 3 positive sodium ions out of the cell for every 2 positive potassium ions it drags back in. This unequal swap perfectly balances the passive forces of diffusion, keeping us locked right at that crucial resting potential of -70 millivolts.
Which brings us to Section 5, the action potential. Our hardware is primed and perfectly balanced at -70 millivolts. So, what happens when a signal is actually sent? How do we actually trigger an electrical impulse? Let's say you touch a hot stove. That initial stimulus interacts with the cell membrane, exposing it to a chemical or physical distortion. Normally at rest, most of the membrane's gated channels are shut tight. But a stimulus pops specific sodium channels open, letting positively charged sodium ions rush into the cell. This causes a localized spike in the voltage, shifting it closer to zero. We call this local, temporary change a graded potential. And the change in voltage is directly proportional to how strong the stimulus was. But here's the catch. A graded potential all by itself isn't strong enough to travel all the way up your arm to your brain. To send a long-distance message, that graded potential has to trigger an action potential, which happens in a very precise sequence. First, that initial localized stimulus has to be strong enough to depolarize the membrane from -70 up to a threshold of about -60 millivolts. This follows the all-or-none principle. It's exactly like flipping a light switch. Once you hit that threshold, the action potential triggers identically every single time. Boom. Step 2, voltage-regulated sodium channels fly open. Sodium rapidly rushes in, and the inside of the membrane flips from negative to positive. Step 3, those sodium channels slam shut and potassium channels open to let potassium out, which repolarizes the cell. Finally, the cell enters a brief refractory period where it resets and won't respond to a new stimulus while restoring its resting potential. That rapid cascading wave of depolarization, that is the electrical impulse cruising down the axon.
Section 6. The synapse connection. Let's zoom back out just a touch and see where that lightning-fast electrical impulse actually ends up.
The action potential travels all the way down the axon until it reaches this expanded area at the very end called the synaptic knob. This is the synapse. It's the critical intersection where the presynaptic cell, the neuron sending the message, has to talk to the postsynaptic cell, the one receiving it. But there's a major problem here. The cells don't actually touch. There's a tiny physical gap separating them called the synaptic cleft, and an electrical impulse cannot just jump across empty space. So, to cross that gap, the electrical software has to translate into chemical messengers called neurotransmitters. When the action potential hits the synaptic knob, it forces these little synaptic vesicles to dump neurotransmitters right into the synaptic cleft. These chemicals drift across the gap, bind to receptors on the other side, and spark a brand new graded potential in the next cell. The neurotransmitters are then quickly broken down by enzymes and recycled, ready for the next signal. It's just a breathtakingly fast sequence: electrical impulse, chemical release, and new electrical impulse, seamlessly completing the path of a single thought or motor command.
Which brings us to a final thought I want to leave you with. Every single memory you hold, every muscle you move, and every sensation you feel is entirely dependent on this elegant integration of macrobiological hardware and microscopic electrical software. Right now, as you're listening to this, millions of your neurons are perfectly balanced at -70 millivolts, actively pumping ions, waiting for the exact stimulus needed to fire an all-or-none electrical cascade across a chemical synapse. So what complex human actions will your -70 millivolt charge trigger next? Thanks for joining this explainer, and keep questioning the incredible mechanics of the world inside you.